Okay, thanks. I just almost finished. I just wanted to emphasize a couple of things. So I was trying to get you to really think about that to turn on a gene, to activate your DNA, the molecules, the enzymes, the energy molecules have to touch it. So it's one thing to diffuse into the nucleus. It's another thing to actually touch the DNA that's exposed. The last slide is just a computer model of that same kind of picture there. Where you can see how it's being exposed. But yeah, the thing I wanted to go over with you here is to get them to touch. In chemistry, they maybe you learned this, but we just say things react because the molecules collide, and there's this what we call the collision model. And what that means is for molecules, they have to collide with enough energy. And secondly, they have to be properly aligned when they collide. And that's really what enzymes or catalysts do for reactions. Remember, enzymes are just catalyst in a cell, a biological catalyst. They're not really part of the reaction. They're not reactants. They're not products. But they set up an environment where these molecular collisions will occur with enough energy, and the molecules will be aligned properly. That's why I really like saying that enzymes set up or create an environment where this can happen. So enzymes can create an environment where this DNA is exposed and allow more enzymes, more energy molecules to get in and touch. Now, a couple things as far as loosening or allowing the DNA to be exposed. We can add these organic acids, these acetyl groups to it. They can cause the DNA to be exposed and really cause the genes to be turned on. Now, turning them off. And once again, this is kind of a macroscopic level because we can actually see this. To turn them off, it's called imprinting of genes. So gene imprinting is turning off. Let me say that again. Gene imprinting is turning off. That also means you're adding these methyl groups. So these parts that are tightly coiled, that are in the center, that are all balled up there. Maybe if you look at the computer model, these parts up here that are all tightly coiled, still all wound up in their protein. Those are turned off. It's fair to think most of those have been imprinted. Most of those genes have been methylated. So once again, this idea of gene regulation is a big topic in this course and a big topic in biology. But right now you kind of have the idea that the DNA has to be exposed to be turned on. We can expose it by adding these acetyl groups to the chromatin, to that protein DNA complex. And the other way that we can regulate genes is we'll kind of look at what turns on those promoter regions. So these parts sticking out probably have a lot of their promoter regions need to be activated or turned on in order for that gene to be transcribed as well. Hey, thanks so much. If you made it through, I think you're in great shape for the quiz. Appreciate all your time.